Hi class, and welcome to the screencast on prokaryotic gene expression. So we've already looked at molecular genetics and the whole central dogma of DNA to RNA to protein. We're looking at transcription and translation, and we're going to have to use this as a foundation for the rest of this unit because we're just going to build on these ideas and it's only going to get a little bit harder. So this is the central dogma that we all remember. Now there is one tiny exception to this that we are going to look at the very end of this video. And so today we're going to look at prokaryotic gene expression, how they can get variation in their genome. We're going to look at operons, and then finally we'll look at viruses. And then at the next video we'll look at eukaryotic gene expression. So we just want to continually go back to this idea that when we process genetic information, it's not perfect, mistakes do happen, and this is a good source of genetic variation. And always remember that changes in genotype lead to changes in phenotype. That is a huge theme for the rest of this unit, especially in this video. And increasing genetic variation is another huge theme, and I'm going to always come back to these two ideas. Why is variation so important? Jot down in your notes right now why variation is so important and why we're going to look at all of these multiple ways that organisms can create variation in their genes. So we're going to look at how are bacteria different than eukaryotes, look at genetic variation across systems. So eukaryotes do it differently than prokaryotes, and then we'll look more at further details in bacteria and in viruses. So prokaryotes are different because their chromosomes are different. They have one circular chromosome as opposed to eukaryotes, which have these multiple linear chromosomes. Remember, humans have 23 pairs. Prokaryotes, viruses, and some eukaryotes can also contain plasmids. Plasmids are these very small extra chromosomal, so they reside outside of the normal circular chromosome. They're double-stranded DNA molecules. So here's a very generic bacterial cell. We have our single circular chromosome, and we've got extra chromosomal plasmids. Now, something really cool about these plasmids is they can often contain beneficial genes like antibiotic resistance, and they're often used as vectors for genes, so they can carry genes along them into various organisms in biotechnology. Now, remember, prokaryotes have no true nucleus, so this region here is simply called the nucleoid region, but it's not separate from the rest of the cell. So let's take a look at transcription and translation. So in this cell here, we have a nucleus where the DNA replicates, transcription happens, and RNA processing happens. And then in the cytoplasm is where translation happens. Over here, all of this is happening at the same time and in the same location. So which cell is which, do you think? Well, hopefully you got it right. The one on the left is a eukaryote because it has a nucleus, and the one on the right is a prokaryote. It does not have a nucleus. So all of this is happening at the same time, and because there's no nucleus, there's also no RNA processing. So this is an important difference between eukaryotes and prokaryotes. All of this is happening simultaneously. Okay, how do we get genetic variation? We already spent a lot of time looking at eukaryotes and how they get genetic variation, especially in meiosis. So jot down those three ways. Remember, two of them happened during meiosis, and one of them actually happened after meiosis. Now, in prokaryotes, there are three distinct modes of variation, and we're going to look at those in a second. But down here, this third one is so important to keep in mind. I'm not going to mention it the rest of the video, so jot it down. Mutation, right? It's an imperfect transmission of genetic information. Mutation happens all the time. This is actually the ultimate source of all variation on the earth and all of life, mutation. All right, but let's take a look at these three modes for prokaryotes. So the first is transformation. This is simply the uptake of naked foreign DNA from the environment from a bacterial cell. So we have to first poke some holes in that bacterial cell, and then it's going to uptake this foreign DNA. It could be fragments, or it could take up a plasmid, and then that DNA can be incorporated into the genome, or that plasmid can just be stably incorporated into the cell. So that's transformation, uptake of foreign DNA from the environment. The second one's a little bit more complicated, and this involves a bacteriophage. Now remember, bacteriophage from the Hershey and Chase experiment is a virus that only infects bacteria. And so here we have the bacteriophage injects its viral DNA in purple into its host cell. Now what happens is it sort of incorporates in with the green bacterial DNA as well. So then when new viruses are packaged, and we'll look at how that happens later in the video, this one little virus here actually packages both a little bit of post-bacterial DNA and a little bit of the viral DNA. So see how it's got both green and purple. Well, then this guy is going to actually go and infect another bacteria, a different host cell, and it's going to inject this little recombination of both. And so now this new host cell actually has a combination. It has a mixture of DNA. And so now this bacterial host has more variety because it got some like new DNA from that virus. So this is transduction and involves a bacteriophage giving some new information to a new host cell. And the last one is conjugation. I just like to think of this as bacterial sex because there is physical contact between the two cells. So here we have on the left side a donor cell. The donor cell is the only one that can create this sex pilus here. So it's just this sort of protrusion, this little bridge that connects the two cells. And then the donor cell is going to be able to pass DNA through this bridge, through this pilus, into the recipient cell. You might hear something called the F plasmid. This F plasmid is always going to distinguish the donor cell, and then once it gives the DNA, the new cell is now going to also become a donor because it has the F plasmid. You basically need that F plasmid in order to make the sex pilus. So those are the three ways that bacterial cells can get new DNA: transformation, transduction, and conjugation, and that gives them variation because remember, they don't undergo meiosis. They undergo binary fission. They're asexually reproducing, so they're either going to do it through these three ways or they're going to get variation through mutation. Okay, now moving on into gene regulation of prokaryotes, looking at the operon. Now this is going to get a little difficult, but remember as we go through these two things, changes in genotype, whether it's an actual change in a gene or whether it's a change in expression of a gene, that's going to lead to a change in phenotype. And second, and this goes back way to early on in the year, all biological systems are dependent and interact with their environment. So we cannot forget how important that environment is to gene regulation and gene expression. All right, so first, a general operon. What is it? Well, it's a functioning unit, a whole unit of genomic DNA that contains an entire cluster of genes under the control of a single regulatory signal or promoter. Now, I want you to draw this in your notes. So over here at the far left, upstream of the promoter, we have regulatory genes. Now, what do regulatory genes do? They create regulatory proteins, which could be, depending on the operon, either an inducer or a repressor protein. Now, these inducer or repressor proteins are going to sit right on top of the operator. Now, whether or not they're bound to the operator is going to depend on the operon and depend on the environment, but that's where they're going to sit. The promoter, remember, way back to transcription, is where RNA polymerase binds and then starts to transcribe. So please, I should have put this on here. I took it for granted. What we're looking at here, this is DNA. Okay, so RNA polymerase is going to sit on the DNA to transcribe it. Now down here, these structural genes, this is what we're actually transcribing. This is what is making the protein or making the enzyme that that bacterial cell needs. And remember, this is only in bacteria that we see operons, not really in eukaryotes. So let's take a look. We're going to look at so I want you to draw this in your notes, so make sure you have all that labeled and explained in your notes. Now, there are two types of operons, an inducible and a repressible. In the video today, I'm only going to look at one inducible because I don't want to overwhelm you. We will look at a repressible in class. So an inducible is normally off, but it can be induced, so it can be turned on. And the example is the lac operon, which we will look at. A repressible is normally on, but it can be repressed, so it can be turned off by a repressor. And the trip operon is an example of that we will look at in class. So let's take a look at the lac operon. Go ahead and draw this in your notes. You can even label it with the numbers that might be helpful for you. Remember, this is inducible. So we're just going to go through and look at each of these components. So 6, 7, and 8, remember, these are your structural genes. These are what actually make the protein that we're transcribing. So these, in the lac operon, make the enzymes that digest lactose. So when a bacteria eats lactose, it's going to need enzymes to digest it. So that's what the structural genes do. Number 3, that's your promoter. Remember, that's where RNA polymerase binds. And then 4 is your operator. Remember, that's where either the inducer or the repressor will bind. In this case, the yellow is the RNA polymerase, and the green, that's a repressor protein. So remember, this is an inducible operon, so normally it's off. What you see now is the off position, but it can be induced to be turned on. So what we see here, okay, look at RNA polymerase. It's not moving, is it? It's blocked. The repressor protein is blocking it. Therefore, there is no transcription. So this is off. There's no lactose in the environment. The bacteria has no need to make these enzymes. It's not eating lactose, so it doesn't need the enzymes to digest it. So it's not going to waste the energy to make these enzymes when it's not having lactose in the environment. So it doesn't need to break lactose down. But let's go ahead and add lactose to the environment. So here in white, this is allolactose, which is very similar to lactose. This is the inducer. So now we have lactose in the environment. It's going to induce the operon to get turned on. So what it does is the inducer binds to the repressor. It changes its shape so that it can no longer bind the operator. And look what happens. Now the RNA polymerase is no longer blocked and it can transcribe those structural genes. So now it can move along, it can make the enzymes that need to digest the lactose. So that bacteria ingest the lactose, it needs the enzymes, it needs to digest it, so it better be able to make these enzymes. And that's just really the basis of an operon. The operon is going to be off when the bacteria doesn't need it, and it's going to be on when it does need it. So it's very dependent on the environment. All living systems, especially bacteria, respond instantaneously to their environment. So enzymes are made. So now it's on, there is lactose in the environment, the enzymes need to be made. Okay, we're going to go really quickly through viral genetics. So we're going to look at how viruses get genetic variation and how they can even introduce genetic variation into their hosts, which we already saw that earlier with transduction, remember? Okay, so just some very basics about viruses. Viruses are actually non-living because they cannot reproduce on their own. They require a host cell. So what they do, and we just saw this, they inject their DNA into the host cell, and then that DNA is going to take over the cell machinery, and it's going to use the host cell machinery in order to make more virus. So here's just a figure that shows this. So here's the virus. It goes into the host cell. It uses the host machinery, transcription, translation, endoplasmic reticulum, to make more virus, and then it can leave. It is released. Now, some important things about viruses. They're highly efficient at replicating, which is very good for them because they can rapidly evolve and change their genotype, which, remember, changes their phenotype. They replicate via a component assembly model. So what happens is they have these very simple components. They use the host cell machinery to assemble all of it, and then it gets released. And replication allows for mutations because it uses the host pathways. And remember, the host pathway can also undergo mutations as well. Now, mutations are especially common in RNA viruses because they do not have a proofreading mechanism. Now, I want you to write down why you think that is. Why do RNA viruses not have a proofreading mechanism? Think about what we learned about DNA and DNA mutations. Okay, simple viral structure. The capsid is the outside, and that's a protein coat. And then inside, you have your nucleic acid. Now, it could be RNA or it could be DNA. It could be single-stranded or it could be double-stranded. There's a lot of variety in viruses. Now, there are two different life cycles, the lytic cycle and the lysogenic cycle. The lytic cycle up here is where the virus gets inside, and it actually destroys the host right away. So new viruses are made, and they actually lyse the cell, burst the cell open right away. Now compare that to the lysogenic cycle where the viral DNA gets actually incorporated into the host genome, and it can remain in this lysogenic stage for years, or it can turn into the lytic cycle. Now with the lysogenic cycle, something really cool is because it's in the host genome, it can actually give that host cell new phenotypes, which could even mean increased pathogenicity in bacterial cells. Both of these viral life cycles have very important functions, though. Facilitated transfer of genetic information. So from parent virus to offspring virus, and from the host virus to or from the original virus to the host cell, whether that be a human cell or a bacterial cell. But these hosts can get new genetic material from the virus via transduction. Also, the virus itself can actually get genetic variation. If two related viruses, and this is pretty cool, combine in the same cell, they can actually combine information and get variation that way. And obviously, remember, as always, a virus can get variation through mutation. I'm going to skip through this because I want to talk about HIV. HIV is a special case. It can undergo rapid evolution, And I'm going to skip through this as well. And it's a retrovirus. Now, this is the exception I was talking about. Instead of DNA to RNA to protein, it actually starts with RNA and then uses a special enzyme called reverse transcriptase to go from RNA to DNA. And then the rest of it follows as usual, DNA to RNA to RNA to protein. So when we fight HIV, a big thing that we actually target is this reverse transcriptase enzyme. Because if it can't use this to get to DNA, the virus cannot work. Now I want you to answer these questions in your notes.